Today I'm going to talk to you a little bit about a set of translocations that we did over from about 2000 up to 2014. And it's a paper that we just released and was published in the Wildlife Society Bulletin. You can see the citation there. A few of our co-authors are in the audience today, but so I don't want to take credit for all of it. I didn't do all the work, but we did pull together all of the data. And also all of the really good photos in here are not mine. They're someone else's. I'd like to thank those folks for those photographs as well. I would like to mention though that there is a point here where we have an AV and the IT people and everything that we're doing. When I gave my first talk 40 years ago, most of us were running around with our slide carols and our slides, and we're running around protecting those for their dear lives. They didn't all fall out. So if you don't know what a slide and a slide carol is, look to somebody at your table that has gray hair, and I'll bet they know what a slide carol is. So anyway, we'll get started talking about survival rates at translocated Gunnison, Sage-Grouse. And if you would like this publication, it's available online at the Wildlife Society Bulletin. Okay. My presentation outline today, I'm going to talk about, in the introduction, a little bit about reintroduction biology. There's a whole science that's been developed here. And then translocation background, basically what the background of translocations are, why do we do them, what are the objectives are. I'm going to briefly cover that. Then I'm going to talk about the study area for our project and the source and recipient populations that you'll see. You've seen them all in Kathy's talk already. And then the methods. I'll talk about the timeline of the translocations, the capture, the marking, the transport, and the monitoring of the birds that was done, and then the statistical analysis. I'm not going to get heavy into our models. It's too early in the morning to talk about AIC values or anything like that, so we're going to talk about survival rates for the most part. And then, but primarily we're going to talk about the number of birds that we translocated, so you get a feel for that and how they're broken down, daily survival and annual survival. And we'll have some discussion and management conclusions for you. Let's see here. Okay. There we go. Okay. Oh boy. Okay. Anyway, usually this happened fast. The source population we take the birds from, as Kathy mentioned, and it's used to establish populations many times, establish new populations, reestablish populations in historic areas, to supplement populations that are existing right now, and that's what we did with Gunnison. And there are lots of other reasons. And then the species that have been translocated over the world have ranged from amphibians and reptiles to our favorites, birds, right? And I put this animal down in the bottom right corner for Brian, just so that animal's there, so we know that it's there as well. So large animals have been translocated as well. This has started a whole group of texts, and people are defining all sorts of ways. Reintroduction biology is a more recent book that was written, but it's also generated a lot of groups and coming up with guidelines for translocations from countries. U.S. Fish and Wildlife Service has one. IUCN has one as well. So it's just it's a new market in many ways. It's been going on for a lot of years. So why do we translocate individuals? Well, usually you've got a population declining, right? You want to reverse that trend. You want to somehow reverse that. You can do that by adding animals, right? You can just add animals to the population, or you may have reasons there ahead of you that there's a genetic bottleneck and you want to add animals to breed and refine the genetics a bit. So there's lots of reasons why you would want to translocate those individuals, but usually to recover something. Here's, you've seen the range of greater and Gunnison sage-grouse. There have been translocations going on with greater and Gunnison sage-grouse since 1933, but there's not a lot of data except we went out and caught about 60 birds and we hauled them into Wyoming, right? That's what you get for the most part. But in the more recent studies, we've got some information. But so to look at survival rates over history, you can see with these red crosses on here, there have been birds translocated across the entire range of Gunnison and greater sage-grouse. So every state, I couldn't find anything for South Dakota, and I certainly couldn't find anything for Arizona, so there's only 2 states. But it was really reviewed well by Reese and Conley years ago, the translocations up to that point. So here's a bar graph for you to look at greater sage-grouse female survival. I'm really just focused here on females because that's one of the better data is. So we do have some data on translocated birds. These are the means of the annual survival rate, which is on the y-axis, is the annual survival rate from 0 to 1, and then each bar is a different translocation. And here are the annual survival rates and then the plus or minus standard errors with those. All the purple bars to the right are the Taylor et al. Paper, which gives us an idea of resident greater sage-grouse female survival. We've got adults on the left and subadults on the right. And then these are the survival rates. The green bars in the first 4 are all from Utah. There were some supplementary papers, and it was real difficult to pull out which one was which, but they were all published papers. And then there's 2 years of data from Washington, and that's the next 2 bars, and then female survival rates for California or for Washington. The big point here I want to make is those survival rates aren't too bad overall, in my opinion. There are some low ones, but they survive much better than many of us thought. They're really close for greater sage-grouse. Colorado has its own history of translocations with sage-grouse. 71, 72, between 30 and 32 birds were moved from the Gunnison. And I have sage-grouse up there because that's what they moved at the time, were sage-grouse. In 92, population estimate was about 50 breeding birds. And then in 2000, when I first came to the state, we had a population estimate of about 6 breeding birds, 1 male and 5 females. So I was directed as my first job was to get down here, convince the Gunnison working group that we needed to take some of their birds and haul them to Poncha Pass, and that's what we did. And so to 2000 and 2002, we moved 51 birds, 29 males and 22 females. So our study objectives for this project were to assemble all the past translocation data and the monitoring data between 2000 and 2014. We wanted to estimate the post-release survival by age and sex cohorts during the first year. We also wanted to compare survival immediately post-release and then after the movement stabilized. We wanted to estimate post-translocation survival by the timing of the translocation, primarily by sex and age cohorts during the spring and the fall. We wanted to evaluate the translocation influence on annual let counts. And then last, we wanted to compare the survival between Gunnison sage-grouse fitted with different transmitter types, primarily rump, mount, and necklace. We've known from some work that Pete's done and others in the West, we're seeing some survival issues with birds that have been marked with rump or necklace mounts, especially the males. And we only focused on males in this one. Here's a study area. You all recognize that study area. Gunnison, of course, was our source population. 2002 to 2002, the birds went to Poncha Pass. Then 11 to 13, birds went to Crawford. We translocated birds to Crawford. In 2010 to 13, birds were translocated to Pinyon Mesa. And then 5 years, differing years, from 7 to 14, birds were translocated to San Miguel Basin. And then in 7 and 9, birds were translocated to Dove Creek. Our methods were pretty basic. Capture birds on the spring, on the leks, at night, spotlighting. Any of you have done that, you know the routine. But we also used, 1 year we used some walk-in traps. And in the fall, we caught birds, sometimes hens and broods, sometimes adult males and females. So they were used the same spotlighting techniques. All the birds were marked either with necklace or rump mount transmitters, and we also marked each bird individually with aluminum leg band or bicolor leg bands in some of the studies. How do we transport them? We put them in a transport box or in a pet carrier. Most of them were in the transport box though. And then we transported them to site with a pickup, an ATV, or a Pinyon Mesa with a snow machine because the season's a little delayed up there. How do we release them? Usually within the next morning after they were captured and transported. At sunrise, the transport box was put out on or near the lek. It was opened, but then also the pet porter was also put out there and the person would open the pet porter and let them out. I consider the transport box, a little more of a soft release, where the pet porter was a little harder release, where the transport box, we'd back off with the rope, slowly open the front, the birds would walk out or fly out at their own pace. They would move at their own pace. So that was a little more of a soft release as far as I was concerned. Within monitoring, birds were monitored on the ground via radio telemetry or from the air or with GPS. And primarily for this study, there was a lot of other information collected by the folks, the PIs at each one of the projects, but we were mostly interested, are you alive or are you dead? And it was a location where you're alive or you're dead. And so that was what we were doing with the survival piece. For the let counts, we didn't do really any rigorous statistical analysis of the let counts. We just did kind of a empirical look at the let counts. because we just didn't feel the LEC counts were sensitive enough to detect the numbers of birds that we were releasing into the population. So I'll show you those figures, but generally we didn't do a real rigorous look there. Within the data analysis, we compiled the translocation telemetry data from all the studies into one file. We censored any individuals that dropped collars or died within 48 hours of capture. We used program mark the nest survival analysis to estimate the daily survival rate, and you'll see that in a minute. And the covariates in our model were post-release, and I'll get into this a little bit, and then population, sex, male or female, the age, juvenile, yearling or subadult, and adult, the season of the release, either fall or spring, and any interesting interactions that we thought were of value, and then the transmitter type. And that was kind of a separate model set that we looked at, necklace versus rump mount. So those were the covariates that went into our model, our daily survival model. So I wanted to mention you notice there where there was a breakdown in the release period from 1 to 75 days and then 76 to 365. And the question I would have is, where did you come up with that number 75 days? And this is where we came up with it. It's in a publication that we published early last year that's available if you'd like it. It was published in Wildlife Research that talks about, we developed resource selection functions for the satellite populations. And to do that, our sample sizes were small enough in the small populations for locations for the resource selection function. We wanted to include translocated birds in that. But we know that translocated birds move a lot very early on, and we wanted them to simulate as much as possible those movements of the resident birds. So the graph you see in the bottom right, I'll zoom in on that here in a second, is the blue line is the resident birds, the red line is regression for the translocated birds, and where those two lines cross, if you have on the y-axis the distance moved per day and the days since the first record, whether it be capture and release or capture and transplant, where those two lines intersected for all the populations. In this situation, you see it turns out to be about 65 days. But in all the populations considered, that turned out to be about 75 days. So where those two lines intersect is where we considered they started moving more like their resident counterparts. Here are some results of the captures. It's the same figure, all three places, but just a little different data in each one. There were more birds translocated than 309, but in the survival analysis, we used 309 birds, and you can see the numbers that we translocated to each population. But what I want you to focus on is up in the upper left, the total number of birds is on the y-axis, and each population is in the bars. The total number of birds translocated, or in the survival analysis, is the total, and it's a stacked bar graph. The females are the green, the males are blue, and then the gray is unknown. In the top right was the season that we translocated them. The season is, the spring is green, and the rust color is the fall translocation. So you see we get a pretty good mix of all of them, which, as those of you know, that's going to start affecting sample sizes, and you'll see that. But the fall, if you'll notice in the Crawford population, which is the far left bar, there were no birds translocated that we included in the Crawford population analysis. Then on the bottom is the age ratio. Adults are blue, yearlings are gold, juveniles are green, and unknown ages are gray. You can't really see that gray on the slide, but it's there. You notice that there were unknown ages and sexes, and we did a preliminary analysis to the data set and to take a look at it and to see which of these groupings these unknowns fell into. And the sex that they best fell into were males for the unknown birds, for the unknown sexes, and the unknown age were juveniles and yearlings. So we then allocated those birds to those groups. So what were the most important factors or covariates in our model? Pretty much all of them. Day 1 to 75 versus 76 to 365 was important to our model. So was population, which really starts to impact things. The sex, which was not a big surprise to us, is that sex was an important part of our model, and age as well. That wasn't really surprising to us. A season was a bit surprising, but a caveat to season is that it was a very marginal impact. Confidence intervals overlap 0, and we had some major issues with that. So it's there, but it was a marginal impact or marginal effect. So this is a graph of the daily survival rates. On the x-axis is the populations, and then these are just males and female adults. But what I want you to focus on, and then on the y-axis, is the daily survival rate, ranges from 0.98 to 0.99. What I want you to focus on is the green bars, and the green bars are the 1 to 75 day daily survival rate, and the gold bar is the 76 to 365 survival rate. And you'll notice in every one of the populations and the comparisons, the green bar, or the 1 to 75, had a lower daily survival rate. And that's obviously going to impact your annual survival rates as well. So if you look at, I won't go through all of these, but just show you that trend of the green versus gold across. So we did have lower survival to the 1 to 75. And then if you exponentiate those out, and I'm not going to get into this, so we'll zoom in again, you'll see all those survival curves are about the same. And you look again, it's I'll wait for it to come up. On the left is 1 to 75 days, and then on the right is 76 to 365. But you notice all three of those survival curves are very close, and believe me, the confidence intervals all overlapped. But it's interesting here is the trend that we saw. The subadult females released in the spring had the highest survival rate, where on the right and on the left, the adult males in the fall had the lowest survival rate when we translocated those. So generally, the colors, the survival curves are all exactly the same, both slides, just a little different positioning on the slide. We then took that 1 to 365. We combined them and produced an annual survival rate. And here the slide I wanted to show you is the annual survival rate again is on the y-axis. And then here are just the populations you can see broken down. And then compare the green to the blue. And the green annual survival was higher for females than it was for males. And that was true for all of the populations. So again, female versus male population, the survival was higher for females, the annual survival. Then the same slide, but different color, different focus here I wanted you to look at was the adult versus subadult. And in all the cases, the subadults, the gold bars, were higher than their counterpart, the gold bars, and by population and by age. And there's a lot more data here that I didn't include, but I just wanted to focus on these because we could look at a lot of different scenarios by population. So here's the female annual survival rate comparison of our data compared to the others. Remember that blue and green graph I showed you earlier, the translocated female greater sage-grouse and the range wide. What I wanted to do is compare here the orange bars with the blue and the green. And you can see that many of our survival rates this is annual survival plus or minus a 95% confidence interval here. It's not plus or minus, but it's a 95% confidence interval that's here. Is you notice the female survival for adults and subadults is pretty darn close to greater sage-grouse. There's one that I didn't include on here, and that's Amy Davis's work in the Gunnison Basin. Her survival rate was 0.61, which is, well, it's right about there. So her resident Gunnison sage-grouse survival was right, our translocated survival was right within what Amy found in her research. Here's that just general look at when we introduced birds, when we translocated birds to each population. These are the graphs that Kathy showed you, this 3-year running average, numbers of males in the year. The red arrow shows you where we actually put birds into the population. The diamonds are where, open diamonds are the year that birds were added, and the dark circles, there were no birds added. And then the bottom right is the Gunnison Basin. And the green arrows kind of show when birds were taken from the Gunnison Basin. So you look here, you don't see much of, the thing is you see an increase by each one of the groups of populations, but then you look at Gunnison Basin, and in 15 and 16, the numbers went up as well. So that makes things a little bit confusing when you start looking at this because all of the populations increased and would probably have increased regardless of translocation. But I will mention that Pinyon Mesa in the center left did show kind of the largest amount of response when we brought birds into Pinyon Mesa. Within discussed inclusions, I'll wrap this up. Bird and sex and age were important predictors of survival, and they're just important predictors of survival overall and with translocated birds. Female and adult survival was higher than male and adult survivor, the female and subadult survival. The differences in daily survival rate, there were differences among populations, so it depends on where you take them. Adult male survival at Poncha Pass turned out to be the lowest that we estimated, and then the subadult female survival at Crawford was the highest that we estimated. And so that gives you a big general idea there. Glade Park, interestingly, had the lowest survival, or had low survival, but yet the highest resiliency in the recovery plan. So that was confounding for us. That didn't make some sense, but you can see the graph. It kind of spoke for itself. Glade Park also illustrated a response to LEC counts, which I mentioned. So what you need to do is expect a response about 75 days post-release. And then, because we had to break our sample sizes down to look at these, we need more information on translocated age, sex, and the timing of release. There was no support for transmitter type affecting survival. And then we have evidence, and I think Sarah's going to present some of that tomorrow with genetic, and then we have demographic evidence with nest success and that sort of thing that shows evidence that the translocated birds are contributing to future populations. Within future translocations, we should attempt to capture subadults of males and females. I've caught a lot of birds. I know that's almost impossible to do, but we got to try if we can. Gunnison Basin is currently our only source population, and we need to evaluate the impact to that source population of taking birds. Managers must start to consider other options for translocation, and the 1,000-pound gorilla in the room is captive rearing. It needs to be considered, be talked about, and discussed, but I totally realize and understand that it can be a problematic issue. We have developed the tools for captive rearing. We know we can do it. I'm not saying we should, but it should be talked about. And we need to have more information on the release of captive birds into wild populations. But we have a little bit of information of that with greater sage-grouse. We have a publication where we added chicks to surrogate broods. That's the Thompson et al. Paper. So those technologies have been worked out, those techniques. And then along with Leif Wickman, myself and Leif produced 2 publications on captive rearing techniques for Gunnison sage-grouse. And so those are all available to you too as well to help make those decisions if you choose to. Oop. There we go. Anyway, with that, I will have you hold off on questions and have Kathy come back up, and then we'll take questions after that.
So thank you, Tony, for that very informative talk. And now we're going to talk about the potential for future translocations and where translocations fit into the recovery plan. So augmenting populations through translocations or captive rearing is a priority 1 action in the range-wide conservation, or in the Fish and Wildlife Service recovery plan. In the plan, they actually prioritize 3 populations, Pinyon Mesa, San Miguel, and Crawford, as being the populations that should first receive birds. It also talks about the need to have sufficient suitable habitat and the threats addressed in that population. I'm not going to talk about that aspect. I'm going to keep to the numbers. But the recovery plan also does state that no Gunnison sage-grouse should be removed from the Gunnison Basin if the most recent 3-year high male count is below the target. So to remind you, currently in Gunnison Basin, our high male count is 480 birds. The target in the recovery plan is 752. We are a long ways from reaching that target to be able to remove birds from the Gunnison Basin. Even though we've got those fluctuations that you've seen, it would probably be 7 to 10 years of great years before we actually could reach 752 birds. But the recovery plan also has a caveat. You could remove birds from Gunnison Basin if further analysis was done to ensure no adverse effects to the Gunnison Basin population. So how is this done? Well, it's a population viability analysis is one way to model the potential for removing birds from Gunnison Basin. So CPW has contracted with Phil Miller of the Global Conservation Network. He has worked on both Greater and Gunnison's population viability analyses in the past. He conducted the first PVA for Gunnison sage-grouse that's found in the 2005 Resource Management or Range-Wide Conservation Plan. And he's also helped the Fish and Wildlife Service on their recent PVA that's included in the recovery plan. We have a team going that includes myself, Tony Apa, and a number of the local biologists, Nathan Seward, Dan Newbaum, Evan Phillips, that work in the local populations, both the source and the recipient populations. We also have Whit Blair from Fish and Wildlife Service on the team, and we're currently still trying to build out the rest of the stakeholders that would be involved in this team. So we're still in the very, very beginning stages of this population viability analysis project. So what is a population viability analysis? Well, it explores the interactions between different known factors, and that's where Tony's work comes in with all the very specific survival rates. Those are very important parameters that would be fed into these models. And when you put all these parameters into the model, it helps assess the likelihood that a population will go extinct, or in our case, a flip side, we want it to survive, the probability that population will survive over some period, specific time period, and under particular circumstances. Our particular circumstances are specific to translocations. We'll be modeling both the source population and the recipient populations, asking questions like how many birds can we safely remove from Gunnison Basin? And how many birds do we need to put into a recipient population to increase the viability of that population? It can answer questions about how many years do you need to do a translocation? Do they need to be subsequent years or alternating years? Time of year, like Tony just pointed out, the fall versus spring that the translocations occur. And since we have 3 recipient or potentially recipient populations, how many birds must be released in one population before moving to another? These are all questions that are very, very important that we hope to get some insight from doing the population viability analysis. Does that mean we can do a translocations? Well, if the high male counts come up and we have positive results from the population viability analysis, can we do translocations? There's a lot of other factors that would have to come into play, and a big one is funding. You can't do a multi-year translocation project if you only have secured funding for 2 years. The last thing in the world we'd want is to get 2 years in and then run out of funding. We've not only wasted effort, we've potentially wasted birds. So the very, very earliest that we would be able to do translocations, if all our ducks got in a row, so to speak, would be the fall of 2023. But we still have a lot of planning to do. We'd have to develop guidelines that incorporate the PVA. There's the potential need to look at genetics, so talking to Sarah. We also need to figure out how we are going to monitor the translocated birds, and very importantly, what measure of success will we use? We need to show that the translocations, if we're doing them, are a success or not. Also, a very important aspect of translocations would be outreach and stakeholder support. It'd have to be a very, very large project across many counties, so we're definitely going to need support from a lot of entities. And there's a potential for new methodologies. Artificial insemination has been used, and that's where a female is artificially inseminated before she is released into the recipient population. Now, a lot of females, when they're released, do not have high reproductive rates. They may not mate the first year, or they may actually have infertile eggs. So the hope is that artificial insemination would mitigate for some of these delayed reactions. It's been tried in Colombian sharp-tailed grouse and did not have very good success. And it has also been tried in greater sage-grouse, and when they used, it was used when they moved birds from Wyoming to North Dakota recently. The results are still out on that analysis. The preliminary analyses did not show that much of an uptick in this females nesting compared to the resident birds, but future analyses or further analyses need to be done on that data. Another technique that's being used recently has been brood translocations. As you know, when you release birds into an unfamiliar habitat, They often travel great distances, move around a lot. They have a higher probability of getting killed in that time. And so another idea that's being used is translocating broods. So they're catching the female with the chicks and releasing them all together, with the hopes that they won't make these big movements. They'll stay in the area where they're released, and they'll actually contribute to that recipient population. This has been tried in North Dakota and California, and it's with very, very good results so far. Pete Coates back there is also involved in those projects. So if you have any questions that you want to get into details, you can hit Pete up for that. So Tony talked about a big ape in the room, and this is the big elephant in the room. What if the high male counts don't come up? What if the PVA shows that there's no way we can safely remove birds from Gunnison Basin? Or if we can remove some birds from Gunnison Basin safely, is it enough to actually help this recipient populations? And so that brings up the big question of captive rearing. And as Tony pointed out, we do have some good information. We have some experience with captive rearing, but there would be a lot more discussion that would need to happen before we actually get something like that on the ground. And since would take a long time to get that on the ground, I think it's important that we might be starting those conversations now. So that's wrapping up the population trends and translocations. We do have some additional time, so Tony and I are available for answering any questions on any aspect of this session. Thank you.
A couple of questions at the distance. When birds are translocated, is there a minimum percentage of native grass and chick food plant cover that can predict that the population will survive in that new area?
Tony's coming up for that. You're going to have to repeat the question, please.
Could you repeat that question for me, please?
When birds are translocated, is there a minimum percentage of native grass and chick food plant cover that can predict the translocated population will survive in that area?
Most of them, if the birds existed there, we translocated to the area. Granted, there are some areas that have better quality habitat than others. So we didn't say this area doesn't have a lot of habitat, i.e. Dry Creek Basin, because just 10 years ago, there were several birds, 15 years ago in there. So that needs to be considered in future when we've got maybe that ability. But right now, we just, if birds were resident to the population, we considered that a good potential for release.
Hey, Tony, this is Darren Long with the Forest Service.
Sorry.
So on your discussion, you had males continuing to be translocated, even though they had like a modestly lower survival in those areas. What's the thought behind that? Is that to kind of boost those?
Remember, this analysis wasn't done until all of the translocations were done. So we didn't, if I understand your question right, we didn't adaptively manage partway through the translocations.
Okay, so would that be a recommendation?
But for future, yeah, you ought to consider that.
Okay, thanks.
Can you tell us the difference in percentage of habitat between the upper and lower Gunnison Basin?
I don't have that information. Like when you talk about the percentage of habitat, there's a lot of good habitat in the upper Gunnison Basin, but there's good habitat all over. Maybe that would be a question that could be directed toward Nathan Seward, our local biologist. But I think we might need more clarification on that question.
I have two questions. My first question is, what was the mortality rate of individuals 48 hours after capture? And my second question is, did you look at climate at all, in particular drought, and how that affected translocated individual survival rate?
You may have to repeat it again. What was the first question was, what was the mortality rate initially
Yes.
released?
Yeah.
I don't have those numbers off the top of my head, but it wasn't overly high. I can't think, you could talk to Nate or somebody in the room. We had only a handful that actually died one or two that died in the translocation process, actually within that. But some birds would die, but it was pretty low overall. They did pretty well overall. But in your second question?
And then my second question is, did you look at climate at all, in particular drought, and how that affected
Look at what?
climate?
No, we didn't. No. It's a good question, though, because the climate does vary from Dry Creek Basin all the way to the top of Pinyon Mesa, right? Vastly different. So, yeah, it does vary.
Hi, I'm Julie Keibel, Dolores County Commissioner. Kathy, great presentation, but in Dove Creek, we raise pinto beans, not soybeans. And I just have to give that shout out because we are the pinto bean capital of the world, so that would look really bad. So,
I owe you a drink, Julie.
okay, all right, that'll resolve that one. Secondly, I don't see a lot of variables mentioned in your data analysis. He brought up climate, which I think is a really good one. I was wondering also about predation and invasive plant species. Were those included at all over the years of study?
No, in the population trend graphs, we don't have that information associated with those graphs. Over the next couple days or tomorrow, today, I think the predation talk is going to occur, and then the noxious weed. Hopefully the information in those sessions will help you decide how to tease that out for the Dove Creek population.
Okay, and I just know that a lot of the collared birds in 2007 and 9, they were found. They'd just been killed by hawks, and I think those are important pieces that need to be worked out as we move forward in the implementation process.
Very good point, Julie. We just don't want to just be feeding hawks when we put birds out there.
So, related to that question, could you speak to the issues associated with agriculture in Dove Creek and Monticello?
Well, there's a lot of agriculture in Dove Creek. The birds are using some of those lands. They're using the CRP lands. They're important lands for grouse. There's just not a lot of sagebrush habitat left in the area.
Has that become more significant in the past 10 to 12 years as its populations have sharply declined?
Can you say that again? I know it's hard to read people's questions.
I think you answered it, but has that become more significant, the agricultural effect in the past 10 to 12 years with the decline in populations?
I don't think there's been a lot of land being broken up, traditional sagebrush habitats broken up for croplands recently. I think the conversion occurred years ago. There's been a change in CRP lands. Some lands are coming out of CRP. And like I said, birds are using some of that CRP land. We've been doing a lot of treatments down there, trying to bring some of those habitats back. And it doesn't help that we've had such droughts recently, because it's very hard to get some of those treatments that we've been doing to actually take and succeed. But I think that's going to come up in subsequent sessions.
Jessica Young from Western. Number one, great presentations, incredibly informative, and seeing the data summarized across the years was really, really fantastically done. I appreciated, and I heard Tony use the word "you" a lot in discussing the ultimate decision about things like the elephant in the room and so forth. And I'd just be curious on who, what organizations, entities, and so forth are the "you" in the back of your mind. Thank you.
Is that specific to captive rearing or translocations?
Yeah.
Okay, to start off, translocations are, for the most part, purview of CPW. We have an agreement with the Fish and Wildlife Service, even though the bird is listed and they have the primary management authority. We do have an agreement that allows us to continue things like lek counts and translocations. Certainly, we would not do it without Fish and Wildlife Service being a partner in that. Captive rearing is a little bit different. How to be diplomatic. That is not a decision that CPW can or would make. That is a Fish and Wildlife Service decision. That being said, in fact, to actually, start a captive rearing program where you have pens that are built, specific facilities, that is something that takes the Congress. It takes a congressional act. So that's one of the reasons I say that it would need, if it's going to happen, those discussions need to start now, because that's going to take a while. So that would take a congressional act to actually provide the funding for translocations. So although that is not a CPW or a state of Colorado decision, we would be part of that. And I say diplomatic because whenever I bring it up, the Fish and Wildlife Service always says to me, well, you guys need to request that. And so it's kind of a chicken and egg thing. So they're kind of saying that the state of Colorado would want CPW or the state of Colorado to request that a congressional act be moved forward. So it would take both the state and the Fish and Wildlife Service, but ultimately it would be the Fish and Wildlife Service. They're the ones that run things like the Condor program, the Black-footed ferret. We're partners, but they are the Fish and Wildlife Service programs.
Yeah, Jessica, to refer to my you thing, maybe it was Freudian. That might be something there that on my part. But what I was thinking of generally, Kathy made a much more detailed, but I was thinking as the group, stakeholders, Fish and Wildlife Service, CPW, all of us. When I say you, I'm a researcher. If you want some technical advice on these things, I can provide that. If there's research needs, we can provide that. But I didn't want to say me, I suggest, right? Because I don't make that decision. And that was where I was kind of headed. It was, maybe I was deflecting and maybe on a couch in a psychiatrist's room. There's a whole different thing for you there, but it might be. But that's what I was thinking in the process.
I'll ask you an online question while Bree's walking over. Have there been any observations, either historically or recently, of within season interlock movement of the GSUG?
Yeah, there is evidence of movement between leks. One of the things that we try to do when we're doing lek counts, we don't always just have a single lek as our lek that we're counting. Sometimes it's actually a lek complex, and that takes into account the potential movements between leks. So we do have leks and lek complexes based on the observations and distance and topography, observations of the counters, our biologists, and then the topography and distance. So we do incorporate complexes, which would hopefully not double count. Well, if we're counting complexes, we're not double counting birds because we're doing coordinated counts on those leks. I think that answers that. Question back here.
Yeah. You were talking about there's been evidence of translocations going back into the 30s. Do we have any evidence that like greater sage-grouse were introduced into some of these populations historically before they were species?
I think there were birds hauled from Wyoming to New Mexico, one example. So if that's your question, yeah, there were some birds way back. There were actually, if I remember the study, the citation, there were depredation issues of sage-grouse in Wyoming and alfalfa fields, and they were capturing them, then moved them to New Mexico. So that would have been in Gunnison sage-grouse range. At that point, it would have been graders moved, but there's no evidence that the birds persisted. So there's been some movement around, but I can't think of, somebody may know somebody, I can't think of anything that's been moved into Colorado that came from greater range. So.
Hi, Kathy and Tony.
Pete, you don't even have to ask a question. I'm going to defer the answer to you.
Great talks, very informative. I have a couple of questions. I'll try to, maybe we could get together afterwards and talk a bit more in depth. But I do have a couple of questions specific and then maybe a broader question. Specifically, I really like the threshold of 0 to 75. It seems to be that we're landing on something consistent across multiple species. For example, mountain quail, we've seen that subadult males also within 30 to 40 days had much lower survival probabilities. And with sharp-tailed grouse as well, we're in that 50 to 60 day range. And what we've done in our models is looked at these time-dependent effects, kind of that bifurcation of where it actually occurs using the likelihood of survival on both sides of that to see where the cutoff is. And I really like the idea of using the movement patterns that you did looking at resident to translocated. But did you use the survival information at all to help guide that threshold? So you could compare it through an AIC approach or something to that effect?
We really didn't, Pete, since that was developed in the RSF paper. We didn't take that any further. So we just kind of accepted that, for lack of a better term. We just accepted that date, that timing for this paper.
Okay, so that was basically the only model,
Yeah.
time-dependent model. Great. And then one thing we found, so moving subadults is an excellent, I think, recommendation, because we're seeing those females survive much better, and males for that matter, and on subadult. However, we're seeing very low nest attempt rates for those birds. This is what's brought us to the artificial insemination as well as the brood translocation techniques. And one question that I have you explored some of those options where you're really infusing reproduction rather than making the success of translocation all about survival of those birds, but more about survival of the gametes of those birds, so that you could get native birds on the ground, just going straight to reproduction? I'm curious about that, what your thoughts are there.
I think if I understand your question right, I think here's opinion. This is not science. This is my opinion. There's a lot to be said about a female. The first point is that I was excited to see those survival rates getting those females to the next year. And so then that one would be more likely to breed successfully. And I think there's a lot in my years of work, there's a lot to be said for a female that you release that has a nest that knows the seasonal use patterns. Right? She knows where breeding habitat is, she knows where summer habitat is, and she knows where the winter is, and she can teach, quote unquote, teach her chicks those patterns. And so I guess in some ways I'm not overly disappointed when they don't breed that first year, as long as we can get to the next year, but that does then require repeated releases that you have to follow up with, because not all of them are gonna live, obviously. And that's why I think we focus more on the subadults, because those survival rates were higher, and moving them into the fall, and moving especially females in the fall, and then they have had some time to adjust, get through a winter, and then attend leks in the spring. So does that get at what you were?
Absolutely. The fall, I think, is really the important point,
Okay.
because a lot of female mortality occurs during that spring and summer period, especially late in the summer. And so that fall, they overwinter really well, right? So that's a good point to make. There's a lot of parallels between Gunnison Basin, the subpopulations, your source population. That's the stability graphs that you showed with the oscillations and so forth, and what we have in the bi-state distinct segment, which is a geographically isolated population of graders, but also on a different listing track and status assessment. And we have a monitoring program set up across all the peripheral populations and those source populations to estimate parameters of all your different vital rates, and also use the lek count data in an integrated population model. And then we could run simulations of different management scenarios to get those outcomes. And essentially, moving birds is nothing more than a migration. So you could influence that migration parameter with how many birds to move, and see what your effects on source population are versus your improvements on the peripheral populations and what the net loss is or net gain is across the entire bi-state. So that's something I'd like to talk to you later about, maybe as far as a technique there, because you mentioned the population viability analysis as guiding how many birds and when to take them from that source population that might make sense for you for the Gunnison area.
Yeah, Pete, we are looking towards the integrated population modeling. We provided some of the data that went into the greater sage-grouse graph I showed you for graders. We're trying to get our data into that format so that we can use the integrated population modeling for Gunnison as well and have better estimates of population sizes.